Right. Good afternoon to everyone. Sorry, I got late to come. What about online students? Are done with the question? What about you guys? You need more time? Almost done. Right. Okay, 5 minutes. I will give 5 more minutes. Quickly finish and give. Okay. How long you need? 20 minutes. I can't give now. Quickly write and finish. Otherwise, stay after and write. Okay. Right. Okay. 5 minutes only. Others finish and give. Next week one. Study enzymes. Have to study. No, it's already finished section. Hurry, you just wait after the class, study a little and write and go. It's okay, it's okay. After the class, study and write. She also did like that last week. She has done that. Unnecessary points you have to remove and write it again, okay? Link කරනද? right. අද පළවෙනි සතියේ. Finish. I'm collecting. Your one. Only some mistakes are there, right? Overall, okay. Graphs are okay. This graph, where's this graph? Missing. Yours are right. Prepare my essay clearly, paragraph format like this, okay? You just came, අපි අද ප්‍රොසෙසිස් කරන ඒ ලෙසන් එකේ මම ටිකක් හැබැයි ස්ටාර්ට් කරලා තියෙන්නේ ලාස්ට් වීක් ලෙසන් එක අනිවාර්යෙන් ඊළඟ බලන්න. You had tried me. I didn't know that you are coming, right? I had the previous class and I took time to travel from here. Okay. Good. Unbelievable. ප්‍රශ්න කේස් එක මම බලලා දුන්නා නේද? දුන්නද? Okay. මල්ලද? මල්ලගේ කේස් එකේ ටිකක් අඩුපාඩු තිබ්බා. බලමු මොනවද කියලා. You don't have to describe, just they are asking the characteristics only. And then these equations are written in words. If you want to write anything in equations, you have to write it in words in a paragraph, right? diagram I make a rewrite කරන්න පොඩ්ඩක් ටික දාන්න, right? That's it. Right. Last week we were discussing about photosystems, isn't it? Right. So since there are new students also, I'll be quickly going through what is a photosystem and all that, right? So, right, now those who are in the class, tell me, what is the location of a photosystem? Location? Thylakoid membrane of chloroplast. Right. Each photosystem has two major parts. What are the two major parts, Putin? Light harvesting complex. So here it is the light harvesting complex. And also reaction center complex. Right. What is containing in the light harvesting complex, Putin? Photosynthetic pigments such as chlorophyll A, chlorophyll B, carotene, xanthophyll, isn't it? Different types of photosynthetic pigment molecules are there in the light harvesting complex. Right. In the reaction center, what are located? What are the things present? Special pair of chlorophyll A molecules which are acting as the reaction center. And here it is. What is it? Primary electron acceptor. Huh? And I told you that there are two types of photosystems on the thylakoid membrane. What are the two types? PS1 and PS2. PS1, also known as P700, that is having special pair of chlorophyll A, which is absorbing 700 nanometer light wavelength effectively. PS2, P680, that is having special pair of chlorophyll A molecule, 680 nanometer wavelength is absorbed effectively, right? So this P600, 700, P680 are the reaction center they are talking about, right? Right? Did I discuss what is happening inside the photosystem? Not yet, ne? Did I? Light energy is absorbed, what will happen? No, I didn't discuss at all. Did I? No. I guess I didn't do that part. Just the structure only, no? We didn't do the what is happening inside excitation of electrons. Okay. Right. Then I'll quickly go through what I discussed also about what is happening inside the photosystem also. So what happens? Light energy comes as energy photons. This light energy is absorbed by photosynthetic pigments in the light harvesting complex. What happens to this? Electrons are, this is the nucleus. Electrons in the shells will be excited into higher energy levels by absorbing this light energy photons. Then these electrons will be returning back to the ground level, releasing the energy absorbed. That energy is absorbed by next pigment molecule. From pigment molecule to pigment molecule, what is moving like this? Energy. I'm using blue color to show that energy is moving. So after absorption of energy, what will happen? This results excitation of electrons from this special pair of chlorophyll A as well. Earlier also it happened, but electrons return to the normal level, ground level. But here, electrons are going to higher energy level that is accepted by this primary electron acceptor, right? So since electrons are moving out of this special pair of chlorophyll A molecules, we are telling that it is said to be that the photosystem is excited. The special pair of chlorophyll A is excited. So P700 is absorbing light energy, is becoming P700 plus electron is removed, right? And then P680, what will happen? Absorbing light energy, it's becoming P680 plus electron, right? It's removing electrons. That means this is being oxidized, right? Okay. What is being oxidized? Reaction center is being oxidized by removing of electrons. If anything is removing electrons, as you know in chemistry, it's being oxidized, right? Understood? So this is accepting these electrons. So here electrons are moving up to this, energy is moving. Can you understand? I told that in the book they are telling this as one step without telling what is happening here. They are telling the photosystem is absorbing energy and electrons are excited, and these excited electrons are accepted by primary electron acceptor. Check whether we wrote that concise form, right? What is given in the book. Can I come and see? We wrote that, ne? Okay, no? Understood? Hi. Now we are going to talk about light dependent reaction of photosynthesis. I told that in the photosynthesis there are two major steps. One is light dependent reaction. What is the other step? Calvin cycle or light independent reaction. So write the topic, everyone. Light dependent reaction of photosynthesis. Sure. Right. I'm drawing something. You guys don't need to copy it until I say yes. Just wait and see, okay? I drew photosystems here. The first one is photosystem 2. Second one is PS1. So on the thylakoid membrane of chloroplast, there are so many PS1 and PS2 are available, right? So for the light dependent reaction of photosynthesis, both of these photosystems are involved, right? So light dependent reaction also we are learning two types. One is linear electron flow. We are learning now linear electron flow. Write the topic linear electron flow. Don't draw, just write the topic only. Right. So now look at this one. The light energy is coming to these two photosystems. Not only to this one also light energy is coming as energy photons. So these pigment molecules are absorbing light energy. The light energy is resulting excitation of electrons, and these electrons are accepted by primary electron acceptor here. Agree? Here also similar thing is happening. Energy is moving and it results excitation of electrons. Electrons are accepted by primary electron acceptor here, right? Right. I'm coming back to this particular system. Now the electrons are here. These electrons which are present in primary electron acceptor of PS2 is now going to move through an electron transport chain which is located at low energy levels. That means higher energy to lower energy. Electrons are moving, right? These are being given to the special pair of chlorophyll A molecules in the photosystem 1 reaction center, because now electrons have moved out of these chlorophyll A molecules. In order to fill that, electrons coming from PS2 is there, right? So while this is going from higher energy to lower energy state, they are going to produce ATP here. ADP plus inorganic phosphate will produce ATP. So in the presence of sunlight, production of ADP, what do we call? Which phosphorylation? We learned 3 types of phosphorylation. So which phosphorylation? Photophosphorylation. Photo means sunlight. Using sunlight, production of ATP, we call it as photophosphorylation. So it's happening here now. Okay? Right. So these electrons are now over here. These electrons are also going through electron transport series, and it's given to NADP plus. NADP plus, H plus electrons will produce NADPH. Do you remember NADP plus and NADPH? NADP plus acting as a cofactor, right? Coenzyme. If you can remember in the second lesson, we learned. So there is an enzyme which is catalyzing this reaction. We call it as NADP plus reductase. Understood? Right. I'm coming here now. Now you can understand these electrons are moved out. Now there is the electron requirement. So water is splitting here. Water is hydrolyzed into oxygen plus H pluses plus electrons. So two of these, 1, 2 plus electrons, 4 electrons are given here, right? So these electrons is filling the gap here. Can you understand? Cannot. Right. So this is a linear electron flow. Why did I tell you linear electron flow? Can you see that Z-like shape is there in this electron flow going in a linear pathway? Finally, it's given to NADP plus to reduce it into NADPH. Then other. Right. Draw this first. I know that not fully understood yet. I didn't do anything yet. Just I'm describing a picture. Which electrons were there? These electrons are given to, yeah, this here. These electrons are given to this one in order to reduce that one. When something is taking electrons, it's a reduction. Correct? Here, that these two levels are two different levels, right? PS2 is somewhere below than this one. So, like accordingly. Firstly, draw, then we'll discuss what is there in the book also. Water is also hydrolyzed due to enzyme catalyzed reaction. Remember that, okay? Alright, I'm asking simple questions. I didn't go to the notes yet, okay? Are you done with drawing? Online students? All done? All of you done? Alright. Yeah. Reaction center. Not that. Here there is something in the book also that they are telling that there is a special pair of chlorophyll A molecules now. But at the lesson note that they are sometimes considering this as one as well. Okay? Right? Special pair or like special chlorophyll A molecules or the reaction center. Right? Just remember reaction center. Right? We can't say like twice. Both of them are accepting and electrons are excited. Right? Okay? Just what is actually happening inside is not necessary. Okay? Hurry. Hurry. Listen to my questions now. So, in the linear electron flow, what are the photosystems involved? Photosystem 1 and photosystem 2. What is involved first? Photosystem 2. Secondly, photosystem 1. Then, what is accepting electrons that are excited from PS2? Primary electron acceptor in the PS2. What is accepting electrons excited from PS1? Primary electron acceptor in PS1. Then, what are the products produced in this line electron flow of photosynthesis? Make a list. Sorry, NADPH, ATP, and here oxygen. Do you remember the big equation now? We are writing an equation for photosynthesis. Carbon dioxide plus water will produce C6H12O6, that is glucose, plus oxygen. So here is the place that oxygen is produced. Can you understand? Right? So oxygen is a byproduct of hydrolysis of water here. Right? Right. So then what is neutralizing the excited PS2? What is neutralizing the excited PS2? Electrons coming from hydrolysis of water. What is neutralizing excited PS1? Electrons coming from PS2. Good. And what is the enzyme involved in reduction of NADP plus? NADP plus reductase. Hurry. Let's go and read this first in the book. Come to that section, line electron flow. Right. It says, light absorbed by the photosynthetic pigments and synthesize ATP and NADPH due to the excitation of PS1 and PS2, which are embedded in the thylakoid membrane of chloroplast. That's the definition of line electron flow. The key to this energy transformation is the flow of electrons from one direction through photosystems and the other molecular components built in the thylakoid. This process is called linear electron flow. Do you understand that part? Second part, ne? More flow of electrons in one direction through the photosystems and other molecular components built in thylakoid is linear electron flow. Right? I'll read that paragraph one more time. See if you can understand. Vashishta or Vashishta newcomer is here online. Alright, come to the next paragraph. The striking of photons of light on pigments results in excitation of electrons from the photosystem 2 to higher energy state. The next one, these electrons will be accepted by the primary electron acceptor of PS2. Splitting of water takes place as a result of an enzyme catalyzed reaction and yields oxygen, hydrogen ions, and electrons. Electrons released as a result of hydrolysis may neutralize excited photosystem too. Excited photosystem, there are other ways to write this one. We can say excited PS2 P680 plus excited P680. Any word is correct, okay? If you want, you can write it and keep it there. Any word showing that it is excited. It says next one, now talking about PS1, okay? So, the events happening in the PS2 is over now. Next one, striking of photons of light on the pigments result in excitation of electrons from PS1 to a higher energy state. Excited electrons will be accepted by a primary electron acceptor of PS1. Excited electrons of PS2 at primary electron acceptor of PS2 will pass through an electron transport chain to PS1 and neutralize the excited P700 plus. That's also excited P700, excited PS1, P700 plus, right? Next it says, the energy released due to the passage of electrons from higher energy state to lower energy results in the synthesis of ATP. This is known as photophosphorylation. Excited electrons of PS1 at primary electron acceptor will pass through an electron transport chain and reduce NADP plus and yield NADPH. Reduction of NADP plus is catalyzed by an enzyme called NADP plus reductase. If the plus is not there, please do write. In the exam, they didn't give marks for that one. NADP reductase is not given marks. NADP plus reductase is the correct one. Okay. Hi. Quickly read this section one more time. We are going to an exam question now. Read then, write down the question first. Write down the question only. I'm giving the answer. No, I'm giving. Let's write. Are you done? So this is one of the reason most past paper questions. So you can understand from the sections that you are studying also past questions coming for the examinations, right? So this time also there will be some questions from the sections that you have covered so far. Only thing is to understand the question properly and write the answer. This kind of questions are directly telling now. Some questions are not exactly telling to write line electron flow. Some way that they are telling to write the line electron flow. So if you have not understood the question properly, that is another problem, right? So this is directly asking line electron flow. Write number 1. We'll write this point form today. Number 1, electrons flow through. Electrons flow through. Electrons flow through. PS1 and PS2. These are standard signs. You can use that one, okay? PS1 and PS2. Number 2, and other molecular components, other molecular components, and other molecular components. Point form it right, okay? Number 2, and other molecular components. Next point, embedded in the thylakoid membrane of chloroplast. Embedded in the thylakoid membrane of chloroplast. Embedded in the thylakoid membrane of chloroplast. Right? Number 4, each photosystem has a reaction center complex. Each photosystem has a reaction center complex. Each photosystem has a reaction center complex. Next one, and light harvesting complexes. And light harvesting complexes. Next one, the reaction center complex contains, the reaction center complex contains, the reaction center complex contains a primary electron acceptor. The reaction center complex contains a primary electron acceptor. Because the photosystems are the ones which are involved in linear electron flow, just without telling what is happening, they are telling the background also, right? Otherwise, without knowing the knowledge of a photosystem, we can't say how it is happening, right? Next one, number 7, I have to give. In PS1, chlorophyll molecule. In PS1, chlorophyll molecule. So here they have taken it as 1. PS1, 6, 7, eh? 7, 7, I'm reading 7. Did you miss anything? Just start from 7th and go, we'll see what is missed. 7th one, in PS1, chlorophyll molecule, put a slash and write P700. Chlorophyll molecule, another name is P700, right? P700 absorbs light of which wavelength? 700 nanometer wavelength. 700 nanometer wavelength. Next point. In PS2, point number 8, in PS2, chlorophyll molecule, put a slash and write P680. Absorbs light of. Now you may complete the sentence. Absorb light of what? 680 nanometer wavelength. Next one, number 9. Now I'm talking what is happening in each photosystem, right? Number 9, striking of photons of light. Striking of photons of light. Striking of photons of light. Striking of photons of light on the photosynthetic pigments. On the photosynthetic pigments. Results in the excitation of. Results in the excitation of electrons from PS2 to a higher energy state. PS2 to a higher energy state. Number 10, these electrons are accepted by the primary electron acceptor in how to complete this sentence? PS2. Number 11, splitting of water occurs due to Number 12, an enzyme catalyzed reaction. An enzyme catalyzed reaction. And number 13, which yields oxygen. Which yields oxygen. Which yields oxygen. Next point, H plus. Instead of H plus, you can write hydrogen ions. Just hydrogen will not get marks. O2. Next point, number 15, electrons and electrons and electrons. Number 16. Next one. Number 16, the electrons released as a result of hydrolysis. Electrons released as a result of hydrolysis. Neutralize. Excited. How do we complete this sentence? PS2. Excited PS2. Instead of that, you may write excited P680 or P685 plus, right? One of these is accepted. Number 17, striking of photons of light. Striking of photons of light on pigments results in excitation of electrons. Results excitation of electrons. Complete the sentence. From PS1 to higher energy state. From PS1 to a higher energy state. And now children, tell me the next sentence. These electrons are accepted by primary electron acceptor of PS1. Write that. Right, next one. Excited electrons at PS2 within brackets at primary electron acceptor of PS2 close brackets within brackets at primary electron acceptor of PS2 close bracket, pass through an electron transport chain. Pass through an electron transport chain. Next point, to PS1. To PS1. To PS1. Next point, and neutralize the excited PS1. Neutralize the excited PS1. Right? To neutralize excited PS1 is 20th point, okay? 22, sorry, 21st, ne? Yeah, 21st one is neutralize the excited PS1. 22, energy released due to passage of electrons. Energy released due to passage of electrons from higher energy state to lower energy state. Results in, sorry, high energy to low energy state is 1 point. Next point, results in synthesis of ATP. Results in synthesis of ATP. Put a slash and write photophosphorylation. (24) Excited electrons at PS1. Excited electrons at PS1 within brackets at primary electron acceptor of PS1 close brackets pass through an electron transport chain. Pass through an electron transport chain pass through an electron transport chain and 25th reduce NADP plus. Reduce NADP plus. Next point, to form NADPH. To form NADPH. Last point, which is catalyzed by NADP plus reductase. Which is catalyzed by NADP plus reductase. Right. Take a little time and read this one more time. Draw this one more time, the linear electron flow. We are going to talk about the cyclic electron flow in this same diagram. So we need to draw it again. Quickly draw, don't take a lot of time. No, cyclic electron flow. Light dependent reaction is having two pathways. What is an electron flow? No one would ask a question like that. What is linear electron flow? They would ask. That the second sentence in that one, the flow of electrons toward the one direction inside the photosystems, something like that is there. No. Flow of electrons, the path flow of electrons through for PS1 and PS2. Right. Are you done with it? Right. Now, usual way to give the electrons from PS1 to NADP plus. So in this case, these electrons are not going to go to this production of NADP plus. Instead of that, they will be using an alternative pathway going like a cycle. Can you see there's a cycle? In this cycle, ATP is produced. If the photosynthesis need to make more ATP, this is helping, right? Okay. But it doesn't mean that linear electron flow is stopped. While it is happening, some of the electrons coming to this PS1 primary electron acceptor will be going as a cyclic pathway, right? Then other, they will be going like this. All right. Draw this one and highlight this circle. You can see that no NADPH production in the cyclic electron flow and no oxygen is released in the cyclic electron flow, no splitting of water, isn't it? Yeah. Right. Let's come to the book and let's read about cyclic electron flow. Come there. This occurs in PS1 but not in PS2. Here are some some photoexcited electrons use alternative cyclic pathway. This produces ATP. NADPH is not produced. Oxygen is not released. Can you understand? Right. Shall we write that right there? This occurs in PS1, not in PS2. Right? Number two, here some photoexcited electrons. Here some photoexcited electrons uses alternative cyclic pathway. Alternative cyclic pathway. Next point, this produces ATP. This produces ATP. Next point, NADPH is not produced. NADPH is not produced. Oxygen is not released. Oxygen is not released. Yeah. Right. I'm giving you 2024 A-level question structured essay also. Yeah. Right. Where are the photosystems located in chloroplast? On the thylakoid membrane. State wavelengths of light absorbed by PS1 and PS2. PS1 700 nanometer, PS2 680 nanometer. Harine? The last question. What did you all write? Is this a comparison or asking one side? One side. About what it is asking? Yeah. You have to write only about cyclic electron flow. So what is? Hmm? No need. What did you write? Both will not give wrong for now, but here onwards, if it is asking only one side, you have to write only one side. Right. Then tell me one answer here. Oxygen is not released. Yeah, occurs only in PS2. PS1, yeah, PS1. NADPH is not produced. Here only not release term is used, not production word is used regarding oxygen according to the given sentence in the book they have taken. Instead of this, you may write splitting of water does not occur. Say one of these should be there, not both are getting marks. What else? Is that all? Let me see what they have taken. Occurs in PS1 only. Only word should be there only. And NADPH is not produced. Instead of that, you can say only ATP is produced. Right? One of these can take marks. Here, O2 is not released is one sentence that they have taken. Another one you may write here. That one, splitting of water does not occur. Splitting of water, instead of that you can write hydrolysis also. Write all the answers, right? So in this kind of questions, let's say that 3 is asked, no? You are writing 4 answers, 1 is wrong. What will happen? You are getting minus marks, okay? Don't write wrong answers, write only the number of correct answers, right? Inside 3, they may write wrong answers, that's fine. But if you write more than that, they will consider whether it's correct or wrong, okay? All right. All done? All right. Let's go to the questions now. Go to question section in the tute. Yeah, okay. Go to the structured essay questions in the tute. I need you to do the first two structured essay pages in the question tute at home. Okay, that is a homework. And please mark these MCQs also in the tute. You write them down, then mark it here, right? Okay. This is from photosynthesis first part. You can now do 2019 question number 5. Those who don't have the tute, please collect it from the class. Question number 5. 2017 question number 9. MCQ, right? I'm talking about. 2011-23. yours. Right. Say thank you, sir. Here, next one. 2003-41. 2002-44. 2002-44. Those are homeworks to do at home. We'll quickly go to the light independent reaction or the Calvin cycle of photosynthesis also, right? Hi. Before that, I'll give you 5 to 10 minutes to go through what we discussed so far regarding light dependent reaction, linear electron flow, cyclic electron flow, right? Okay. If you have any questions related to those areas, let me know. Structured question, first 2 pages here. You don't have? I have it. First 2 pages. If you don't have enough space to write answers in the structured essay, better to write it in the writing book and write the answers, right? Okay. Yeah, these 2 and those MCQs. Yeah, having. I'll let you know what content is coming, right? MCQ with Rallium. Hari, හැමෝටම පුළුවන් එතකොට 2 hours MCQ. You can come later, not 12. At 1 o'clock, please come here. Some of you have classes after, ne? You also have 1 hour in between, so write and go to the class. Right? I'll let you know about the procedure in a while. I'll tell you. Right? ගීතන් සර් මේ ළමා දිනේට අයිස්ක්‍රීම් දීලාද කොහෙද මං තනියෙන් කන ඔයගොල්ලෝ දෙන්න මන්නේ ළමයි ඔයගොල්ලෝ ළමයි ඈ next week paper I'll tell you wait and then next week after then next week man. Okay, nama kano tani hobe oborne. Thank you, Putu. All of you wrote this one, eh? Erasing. Mm. Right. Now we are going to Calvin cycle of photosynthesis. So it's a person's name. Calvin is a scientist who has found out many of the reactions in this pathway. So this pathway is named after him. Okay? Right. So now in the photosynthesis reaction, we know that carbon dioxide plus water will give you say 6 H12 or 6 plus oxygen. Now we know oxygen is released, water is utilized, but we don't know where that carbon dioxide is taken and how this is being formed. So in the light independent reaction or the Calvin cycle, we are going to learn how these things are happening, right? Okay. So it's a cycle, no? So this is having events which are happening like a cycle. So the location where it is happening. This is happening in the stroma of the chloroplast. Light dependent reaction happened in the thylakoid membrane of chloroplast, and this is happening in the stroma of the chloroplast, so that you know that enzymes necessary for the Calvin cycles are located in the stroma, right? So the products produced during light dependent reaction are necessary for the Calvin cycle. What products? ATP is needed for the Calvin cycle. NADPH is also needed for the Calvin cycle to occur, right? Understood? Hi. Write the topic Calvin cycle, write the location also. In the book Calvin cycle, there is a big error, children. So I'm correcting that one. Even the professor who makes the paper said that the correct is what I am telling. It's not given what is given in the book, right? There's a little error. I'll change that when I'll tell you the proper thing. And also the reference book is Campbell, right? I'll give you the pages of Campbell also that you can go through and see what I'm saying is correct or wrong. Okay. All done? Erasing. Right. So in the Calvin cycle of photosynthesis, carbon dioxide is accepted. Carbon dioxide, from where the carbon dioxide is getting into the leaf? From the atmosphere. Via stomata, it is entering to the substomatal cavity and then going to the leaf mesophyll cells, right? You can remember the structure of the leaf, no? In all of us, you had a little, ne? For example, if I draw, this is the epidermis, lower epidermis also there. Here are stomata, the mesophyll, palisade mesophyll, and spongy mesophyll. So carbon dioxide is entering through this stomata and going to these mesophyll cells. These two types are mesophyll cells. So the carbon dioxide is now there inside the cell. So now it is going to the stroma of the chloroplast. Now it's going to get captured by compound called RUBP. This is a 5-carbon carbohydrate. So 5-carbon compound, ribulose bisphosphate. Right? You don't have to remember the long term. The short term is enough. It is also accepted. So RUBP is accepting the carbon dioxide. It's producing 6-carbon unstable compound. Unstable compound is not going to stay longer time period, no. So it's going to broke down into 2 molecules of 3-PGA. This is 3-phosphoglycerate. This is a 3-carbon compound. Okay, I'm removing the long terms. You don't need to memorize this. Okay, so this is 5-carbon. This is 1-carbon. 5-carbon plus 1-carbon. You can understand 6-carbon compound is produced. Now it is broken down into 2 molecules of 3-carbon compound. Got it? Right. So from here to here, we call it as carboxylation or carbon dioxide fixation. Right? Here to here. Then 3-PG is converted into 1,3-bisphosphoglycerate using some of the ATP produced in the light-dependent reaction. And it's being converted into G3P, which is glyceraldehyde 3-phosphate, something that you know. How many carbon are there in glyceraldehyde? 3 carbon. It's a 3-carbon compound. This is also 3-carbon compound. This is again a 3-carbon compound, right? So this is using some NADPH produced in the light dependent reaction. It's converted into NADP plus H pluses plus electrons. Understood? So what's happening here? NADPH is converting into NADP plus. That is an oxidation. When this is oxidized, what will happen to the 1,3-bisphosphoglycerate? It's being reduced. I told redox reaction, if one thing is oxidizing, the other one should reduce. So here, this is oxidized, this is reduced, right? So this is the place where the reduction is actually happening, but we are considering from here to here, we take it as one step, 2 molecules of 3-PGA converted into G3P, right? So two of these, balancing I'll do one more time again with the correct numbers. You can understand that considering this as one step, using ATP from light reaction and NADPH from light reaction, 3-PJ is reduced into G3P. That's how your book says, okay? As considering single step. So here to here, we call it as reduction. So this is the second step of Calvin cycle. Then this is the first first carbohydrate precursor. What is the meaning of precursor? Before formation of real carbohydrate, this is the one which is produced. So this is going to produce what compounds? No, glucose. Yeah. So this is going to produce glucose in future. I'll talk about that later. But some of these only going to produce glucose, but remaining will be going to regenerate RUBP in several steps, right? So this is the third step that also utilizes of ATP from light-dependent reaction. So number 3 is regeneration of RUBP, else you can say regeneration of CO2 acceptor. Correct? Up to that, okay? So tell me, what are the 3 steps of this Calvin cycle? Carbon dioxide fixation, reduction, and regeneration. What things from light reaction necessary for Calvin cycle? ATP, NADPH. Right, good. Then what is accepting carbon dioxide? RUBP. How many carbon are there? 5 carbon. This reaction is catalyzed by an enzyme called Rubisco. The long name for Rubisco is RUBP carboxylase oxygenase. Good. This can catalyze 2 types of reaction, oxygenase and carboxylation. Here we are talking about carboxylation for the moment. Right? Right. So the next question is, what is the first stable product produced in this pathway? 3-PGA, this one. Right? Okay. And what is the first carbohydrate precursor? G3P. Understood that? All right. Right. Let's draw this one. I'll be drawing this one more time to show the correct balancing also. Draw this first, then we are drawing next one also, right? Another one also to balance. Reduction. Here, here you can see that NADPH is converted to NADP plus. That means removing electrons, meaning that this compound is oxidized into this to this. While this is oxidizing, this is reduced to this. Actual reduction happens over here, but in the book they have taken whole single step 3PGA to G3P as reduction. That's the correct way. Okay, both ways are correct, but they have omitted this middle step. Sure. Hi. Okay. Right. So G3P is having 3 carbon now, Gutte. So since G3P is having 3 carbon, for glucose, how many carbon are there? 6 carbon. So in order to produce one glucose, 3 G3P should get together, isn't it? So 2G3P. 2G3P will produce one glucose molecule. Agree? Right. They say that for one net production of one G3P molecule, cycle should occur 3 times. We'll balance and see with three times. 3 times means 3 carbon dioxide with 3 RUBP. Agree? Then here 5 into 3, 15. 15 plus 3, 18. How many 6 carbon unstable compounds? 3, no? Neighbor? Right. Then how many 3PG should be produced? 6 of these should be produced. Then 6 of 1,3 phosphoglycerate. Then 1 G3P, which is the net production in the sense G3P, which is going to generate carbohydrates. So 1 G3P is going here for carbohydrate production. That's what they say. So here 6 of these are produced now altogether, out of which 5 are going to regenerate RUBP. Right? If the cycle occurs 3 times, 1 G3P is going to produce carbohydrates and the rest of it will be regenerating RUBP. Now everything is balanced now. Now think and tell me, in order to produce one glucose, how many times this cycle should happen? 6 times. 6 times, isn't it? Understood? Right. Write this down with the balanced numbers, new numbers again, and write a sentence there. Did you write the steps here? Write the steps also. Oh, net synthesis of 1 G3P cycle should occur 3 times. 2 for synthesis of 1 glucose cycle should occur 6 times. For the examination, you are getting microscopes, cellular organelles from that section until the end of meiosis, right? So please study that part and come. Those who have not come to the cellular organelles, that is just simple section. Just read the book and see whether you can write the answers, right? Newcomers. They miss only a microscope, that part. Microscope you miss. MCQ only, 50 MCQs are there. 2 hours. Hmm. You will be able to finish it early. Come at 1 o'clock and do the paper and submit it to the Akis there and go. Yeah, microscope, everything is coming. Okay. Just take a download a picture and see. It's belong to practical side. Enzyme, there's one practical I have to do. Right. Now, everyone, once you are done with this, write these 2 sentences. Yeah, because 15 years of 15 RUBP can regenerate 3 RUBP there. Right. So, Putte, did you write these 2 sentences? This is not that hard. Some children are confused in these facts. Okay, go through. If you still don't understand anything, when I meet you on next class, ask, okay? And do more questions from photosynthesis that will sort a lot of your problems. And those who have done with the picture, please go to the book. I told that there is something wrong in the book. Just read and see what's wrong there. Read the Calvin cycle completely. Is there anyone who is getting late? If I keep you until 3:15, can stay? Okay, no issue. Yeah. That one is correct. The reduction and carbon dioxide fixation, one sentence is above, neither. Yeah. Yeah. Let's go to the first part. Carbon dioxide fixation. Yeah, that's given. They have given up to that only, ne? Hari, it's correct. Is there any book who is having the first part in the carbon dioxide fixation that 1,3-bisphosphoglycerate also involved? 1,3-bisphosphoglycerate in the carbon dioxide fixation part and using ATP or something like that? Because they must have changed it. Here, two different things. This is the first favorite product of photosynthesis. The enzyme RBP carboxylase oxygenase. Up to that only, neither that they have taken this sentence down, neither, and that is removed. Here, this book is the year 17 one, ne? All book, ne? A new book they have added, ne? What is your edition? No, they have not included that in the 2017 version, but they have done some change here, but change is not worked properly, right? Something is wrong here. Here it says, I'll correct it properly. Wait. Open the tutor also, go to the Calvin cycle. In my one also, there's some change to do. All right. Go to the book first. Let's read from the beginning, the Calvin cycle. The Calvin cycle takes place in the stroma of the chloroplast. Energy from ATP and NADPH produced by the like reaction are used to reduce carbon dioxide. The reactions are catalyzed by enzymes and their sequence was discovered by scientist Calvin. This is an anabolic reaction. Product of the Calvin cycle is glyceraldehyde 3-phosphate G3P. For the synthesis of one molecule of G3P, cycle must take place 3 times. That part is correct. Calvin cycle can be described in 3 steps: carboxylation or carbon dioxide fixation, reduction, regeneration of the carbon dioxide acceptor. Come to the carbon dioxide fixation, carbon fixation. The carbon dioxide acceptor is 5-carbon sugar, ribulose bisphosphate. In addition to carbon dioxide, to RUBP is called, so the addition of carbon dioxide to RUBP is called carboxylation. The enzyme involved in this reaction is RUBP carboxylase oxygenase. Okay, that part okay now? Give me 1 minute. Hello? Oh. The next sentence, the first product of RUBP carboxylation is 6-carbon molecule which is unstable and breaks down immediately into 2 molecules of 3-PGA. This is the first stable product of photosynthesis. The rubisco is present in large amounts in the chloroplastoma. Up to that only, that is necessary for the carbon dioxide fixation. Remove that last sentence. 3-PGA is converted to 1,3-bisphosphoglycerate by adding 1 phosphate group from ATP. Put that into the reduction phase. The last sentence, 3PG is converted to 1,3-bisphosphorylate by adding 1 phosphate group from ATP. Last sentence. Yeah, if you don't have, that's fine. Okay. Look at the tutor also. Check that is given properly. If not, put that bullet pointer down. Okay. Hi. Next one, reduction phase. So I'm taking last sentence from that one to this one. 3PG is converted to 1,3-bisphosphoglycerate by adding 1 phosphate group from ATP. So that means this conversion is said there. Next one, 1,3-bisphosphoglycerate will be reduced to glyceraldehyde 3-phosphate step by step enzyme catalyzed reaction utilizing NADPH and ATP from light reaction. Talking about this whole story. Then G3P will act as precursor for carbohydrate synthesis, glucose. Understood that part? Last part, regeneration of RUBP. RUBP is regenerated by undergoing a series of complex reactions. This process uses energy from ATP generated in light reaction. Glucose is later synthesized from G3P through series of enzyme catalyzed reactions. Understood? There are some more questions from this section also in the structured essay area. Please go to that one. Structured essay. In the structured essay, you can do it at home. Question number 13 and 14. Okay, question number 13 and 14. You also can collect tips from the class. Okay. 13 and 14 in the structured essay. You have to do it at home and come. Everyone's answers I'll be marking so that write it properly and come. Yeah. Yeah. What's given there? First product of carbohydrate. First stable product. Okay. No, this is wrong. That sentence is omitted there. Remove. Not G3P, but the 3PG. ඔයාගේ වැරදි. Wrong in here. 3PG. Yeah, yours is correct. This is not the first product. No? This is the precursor of carbohydrates. They had so many errors in this area. You can see that it's not clear. No, some sentences in photosynthesis section you can't even understand. I'm trying to make it clear as much as possible. Still, there may be errors because you know that this concept is all new to you. This you didn't learn in all of us now. So that please refer the book and I will send you pages from this Campbell also. If you want, you can read it and understand more. Okay. Right. So everyone, good afternoon and have a nice day. Next week, exam, MCQ only, 50 MCQs from microscopes to until the end of meiosis. So be ready with that and come. Okay.